So you've had a look at the diagram on page 177, and we'll just go through it very quickly. So A and B are dermal tissue. Here, so you have your cuticle, which is your waterproof layer, and your upper epidermis. And F down here is your lower epidermis, and in your lower epidermis you will have your guard cells, and you will have gaps in the guard cells, which are stoma if it's singular and stomata if it's plural. In terms of our ground tissue then, it is C, D and E. So C are these tall rectangular cells here. This is your palisade layer, and this is where the most photosynthesis happens in your cell. We have then D, which is the spongy mesophyll. So the sponge suggests there are holes, and in here you have air spaces, which suggests that there's a diffusion of gases happening here. And over here, J and I, these are your vascular tissue, and with your xylem on top and your phloem on the bottom. So this is a transverse section of a leaf. You will be asked to draw it or to be able to label its parts. You have these questions on page 177, so you should have an idea at least of these answers to these 4 questions, because we've done them in different parts, whereas transpiration and your understanding of this may be new because it isn't on your junior cycle. So in Form 4 we talked about movement of water, and you should have the name of a process in your head, and plants don't always move water using that process, and you need to know where that process doesn't happen. So autotroph, we saw this when we did the digestive system, and you should be aware by now that plants are autotrophs, and it is a form of nutrition where an organism makes its own food. And why is it that plants need a transport system? Well, it's because they have loads of metabolic processes that are ongoing, and they need to supply substances to different parts of the plant so that these processes can occur. So for photosynthesis, they're going to need things like carbon dioxide and water. For respiration, they're going to need glucose and oxygen. And for growth, they will need to, for cell growth, they're going to have to make organelles. They're going to need substances for that. And for either asexual reproduction or sexual reproduction, again, they're going to need substances for that. So you should be able to answer at least 3 of the 4 of these. So water has to be transported, and we've said that if you talk about water, you need to mention minerals, or at least understand that minerals are come at the same thing. Carbon dioxide has to be moved within a plant, and any of the carbohydrates produced in photosynthesis. Now, we're going to keep it very simple, and as far as we're concerned, the only carbohydrate is going to be glucose, and glucose can obviously then be used directly in respiration, or it can be stored. So we've done starch and we've done cellulose when we did our food biomolecules at the beginning of Form 5. So water enters a plant. Hopefully you're all telling me that water enters a plant at the root, which is correct. Believe me, I've had some junior cycle science tell me that, oh miss, it's the leaves, because water falls in the leaves, so that's how it gets in. Specifically, it's via the root hairs, and we've done the method in Form 4, which is osmosis. So you now all hopefully have a definition for osmosis. So osmosis is the movement of water across a semi-permeable membrane from a region of high water concentration to a region of low water concentration. And when we did osmosis, when it goes from high to low, it's along the concentration gradient. It's not working against it. And we had 2 types of transport, active transport and passive transport. And you should know that osmosis, moving from high to low, is passive transport. No energy is required because it's moving with the concentration gradient. So what's really important to know is that in a plant, if these conditions are met, you have a region of high water and a region of low water, osmosis is going to occur. You can't stop it. So when we did osmosis, I kept telling you that the SEC will ask questions in the context of some region being more concentrated than something else. And you have to recognize that is talking about sugar or salt, and what does that mean for water concentrated. So this is more concentrated, this left side, than the right hand side. And what I've wanted you to recognize, if it's more concentrated in sugar and salt, it means there is less water on this left hand side, which must mean over here there is more water. And if there is more water, then osmosis is going to occur going this direction. So just in terms of this diagram, you've seen it before. This is our semi-permeable membrane. It is going to be a cell membrane in a plant or animal cell, and in our mandatory practical it was visquing tubing, and this is just the water, and this is your glucose or your salt. Now, transpiration is going to be new. So transpiration kind of sounds a little bit like perspiration, and obviously when we perspire we produce sweat. So transpiration, it's not plants sweating, but it just gives you the idea that there's a release of water, is more the hint I want you to take from that. So transpiration is the loss of water vapor by evaporation, and it can occur in all aerial parts of the plant, but most of it occurs through the stomata. So you will get water vapor leaving cells and it'll be released out through your stomata. So you need to have a definition and you need to know where it takes place. So over the page now you have something that looks like this, and these grey boxes should be very obvious for you, and nothing is going to go into these grey boxes. You have a line under water uptake to fill in, and we will fill that in a minute, but first thing we're going to talk about is the adaptations of our roots. So this is a picture of a root, and these little things coming out of it are the root hairs. And what you need to know is that so you have thin walls in these root hairs which will allow substances to move through. So give you the comparison of alveoli, 1 cell thick. This is in the same vein, and the fact that there's so many of them. Root hairs means that there's a really large surface area, so you can get lots of movement. So the adaptation is the root hairs have thin walls with no cuticle, so no waterproof layer, and they have a large surface area. We've done the method, so the method is osmosis, and the sentence to go under your header of water uptake is that the root hairs are extensions of your root epidermal cells. They are part of your dermal tissue in your plant. Now, so we have this. Explain why osmosis works in terms of salt concentration. So somewhere it has to be more concentrated. So where is that? So if you think about your root cells, they are going to be more concentrated in salt and sugar than, or definitely in salt, compared to outside in your soil. So in your soil it's going to be less concentrated, it's going to be less salts, it's more dilute, which means there is more water. So more concentrated up here means there's less water. So water will move from the soil into your root hairs via osmosis. So to explain it in words, the cytoplasm of your root hairs contains dissolved solutes, making it more concentrated. And if you need to put less water, in brackets for yourself, then the water in your soil, which is going to be less concentrated in salts. And for that reason, osmosis will happen. So here now is where it gets weird. So these are our root hairs. This is our ground tissue. This is our transverse section of a root. And how do I know it's a root? First and foremost, there is only one vascular bundle, is the first giveaway, or it's the top answer for the SEC. And the next answer is that there are root hairs. And here is our xylem, and the phloem is out here. So we have said that water is going to be more water outside here, and it is going to move via osmosis into the root hairs. And then here's where it gets weird. So to get through the ground tissue into the xylem, it actually moves, as far as the SEC are concerned, by diffusion from your root hairs into your ground tissue until eventually it reaches your xylem. So the method for into the ground tissue and xylem is by diffusion, and we've already done this when we did our vessels and our tracheids. That xylem is a continuous hollow tube, and it goes from the roots all the way up through the aerial parts of our plant. So now we start talking about, once it's in our xylem, how is it actually getting to go from this point here all the way up to the very top of a tree. And we're looking at this in terms of the ordinary level content first and foremost. And in our next lesson, lesson 2, we'll put the higher level bits to it. So there's 2 methods in which water can move up through the stem in the xylem. And the first thing is a thing called root pressure, so that's our header number 1. And if you wish to push in brackets, so root pressure pushes water up the xylem, and the second one is transpiration, which occurs at our stoma, in our leaves, and this pulls water up the xylem. So in terms of the little bit more detail, the blurb is, as water moves into your roots by osmosis, it builds up a force that pushes water up your xylem. So this is going to be very badly drawn, but you're just going to have to bear with me. So we have said you can't stop osmosis as long as the high water to low water regions are met. So what will happen is, this would be the first layer of water molecules that would come in, and as more water comes in, these guys would move up in the bed. So this is my first layer, and my second layer has now come in, and again water is going to keep on going. So this is my first layer, the second layer is underneath it, because every layer that comes in pushes the other water molecules up. So it's a force that's going to push the water up the xylem, but gravity is working against that. So eventually your root pressure can only push the water up the xylem so far, and then other things have to happen to help it out. So in our little diagram over here of our vessel, you can see the little pink dot, and just, it's a very simplified diagram, but water moves in and it literally pushes it up. Not quite like that, but just to give you an idea. So transpiration, we've said, is loss of water by evaporation through the stomata, and as it does this, as water leaves through your stoma, the next water molecule is going to be pulled after it. So this is going to be my little guard cell down here. Okay, I'm not drawing the detail for the epidermal cells, but the idea being that this is our water molecules, this is going to be our airspace, this might be one of the spongy mesophyll layers, and as this one water molecule comes out, it pulls the next water molecule with it, and so on and so on and so on. So they literally will get whipped away into the atmosphere. So I've got a push and I have a pull. So the push comes from the roots, pressure from entering into the roots, and the pull comes from as water leaves through your stomata by evaporation. So if one gets pulled, the next one goes along with it. And this will make a little bit more sense when we do the higher level part. So just the extra little bits. So we have the push and the pull. In terms of transpiration, because you're getting this loss of water and out through your stoma, you get this what's called an osmotic gradient. Your leaf cells become less turgid, and we're going to talk about that. So this is words we saw when we did movement of water. And because this osmotic gradient is there, water will continue to move out through the leaf unless there is something to stop it. And that something is going to be the fact that your stoma will open or close depending on the conditions. So we talk about water loss in leaves and how do you control it. So this is just an FYI. If there is loads of transpiration ongoing, then water will lose more than their weight in actual water, and if it's not replaced, then the leaves will wilt and die. So this is me and my love affair with basil. So I leave it watered every morning, and if I'm not home in time, then it will have wilted because of transpiration inside my glass window, and it's losing water all the time. So there's 2 main ways that plants control water loss. We've seen one of them. So the first way is through the presence or absence of a cuticle, and its present reduces water loss. There's only water loss through one side of your leaf, the lower epidermis. and obviously the cuticle isn't on the underside of the leaf, so water loss can occur there. There are some plants or some leaves that don't have cuticles, so basil does not have cuticles. And the second method is the opening and closing of your stomata. So at the bottom of your page 179, you can put this little diagram in here. So this is your stoma. If you were looking at the bottom of a leaf when it's open, this is the gap that's there. So again, if we're drawing this, I will come out and draw it for you. So if we are drawing this, we are drawing our cell membrane. We're drawing it curved, we're giving it a nucleus, and as my Form 2 say, we're giving it a swimming pool. So when your stomata are open, your cells are full of water. They are what are called turgid, so they're real chunky monkeys, and this is going to be the gap. So these are the guard cells, and this is the gap, which is the stoma. For your guard cells to actually close, so when we draw it in the longitudinal section, this is how we are drawing it. Obviously this has a seat and a swimming pool. So that's the LS version, and this is just literally looking upwards at the bottom of a leaf. For our guard cells to close, then they actually have to lose water. So they're not as turgid as they were. They are also not plasmalized in that the cell membrane hasn't retracted from the cell wall, but they just don't contain as much water, so their vacuole would be much smaller. So there's now no longer no stoma gap, and this is your guard cell. And again, if we were looking at this through a longitudinal section, then these guys would now be touching. There would be no gap for no transpiration, no gas exchange to occur. So in terms of the words, guard cells open and close by changing their shape. If your stomata are closed, there's no water loss, and if your stomata are opening, well, then your water loss is increasing. Now in terms of this, I just need to move this to one side for you. So again, this is just the view that we have drawn, and you can see that if the cells become turgid here, they swell up, they make the wall bend and your stoma open. And similarly, if they, depending on the conditions, they lose water, so the cells join together and your stoma will close. Now, your stoma are usually open during the day for gas exchange, and they usually close at night. And the reason for that is gas exchange isn't necessary because there's no photosynthesis, so no oxygen is needed, no carbon dioxide is being produced. If you can remember that there's no photosynthesis at night, this will help you to remember what is controlling, actually making your stoma open or close, making them become turgid or making them lose water. And we'll do that in a second. So the rule is they're open during the day, they're closed at night, but actually there are some conditions that will make your stoma open or make them close. So during the day, they will close if your plant is losing too much water, so there mightn't be enough water in the roots. The stomata will close. If the temperatures are too high, it means the rate of evaporation is really, really high, so the transpiration stream from root to stoma is just flying. They will shut up shop. Again, if there's high winds, so the winds will be pulling water out through the stoma, and if it happens too fast, then the stomata close. And if the weather is too dry, so if again there's not enough water in your soil, the stomata will close. So this is the general rule up here. They're open during the day, closed at night, but depending on your weather conditions, they will also open or close. So please be aware of that. Now, over on the next page, this is our last little bit for this section. So stomata, how do they physically open? So we've done little bits of this. So the guard cells curve when they absorb water and they swell, and the gap between the guard cells increases in size. And what is causing them? So that's the physical observation for stoma, but it's your concentration of carbon dioxide that actually determines whether they will open or close at all. So if the concentration of carbon dioxide is low, your stomata open. So we've said if you can relate photosynthesis and what's going on, then that will help you with the reason why they're opening or closing. So why might your concentration of carbon dioxide be low, or where might that be? So if a plant is photosynthesizing, it is using up carbon dioxide. So once daylight comes, your rate of photosynthesis increases, and suddenly there's low levels of carbon dioxide in your air spaces, and to get more carbon dioxide is in, your stoma has to open. So it is your carbon dioxide concentration that determines whether they're going to be opened and closed. So during photosynthesis, low levels of CO2. To get more CO2, the stomata have to open to allow gas exchange to occur. So then similarly in terms of stomata closing. So how do they physically close? Well, the guard cells lose water and shrink in size, and your stoma will close. And they will close when your concentration of carbon dioxide is high. So what has to happen in your air spaces for carbon dioxide to be high? Well, there has to be no photosynthesis happening, and that's going to be at night. There's no light, so there's going to be no photosynthesis. You have now high levels of carbon dioxide in your stoma, and your stoma will close. So we have how they physically open and close, and we have the reasons why they physically open and close. So it has to do with the rate of photosynthesis and the levels of carbon dioxide in your air spaces, in your ground tissue. Now there is an experiment. So if we had been in school, we would have had leaves. We would have been putting nail varnish on the bottom of the leaves, and we would have put sellotape over it and taken the layer off of nail varnish, put it under a microscope slide so that you could see the stoma. And there's an experiment that you can do to prove that it's CO2 is responsible and not light. So if you put your plant in the dark and you put it with a scrubber that would remove CO2, so we would have our dark plant, put it over here. So you literally put your plant in a black bag, you'd have a little scrubber to remove your carbon dioxide. You would obviously have air coming in, and you would have to have air being able to get out, but you'd literally leave your plant in here like so, and what you would find is you put your hands in there, find a leaf, put nail varnish on it, then get your sellotape and put the sellotape on a slide, and when you put it underneath on a microscope, you would see that the stomata are physically open. So it is constant carbon dioxide concentration that determines whether stomata are open or closed, not whether light is present or not. So that brings us to the end of lesson 1. So go have a little break.